Let's have a quick look at the microneuroanatomy. What is the brain made of? Now, we've already been discussing quite a bit of this already, so we've covered some of this territory with the structure of the neurons and so on. So let's look at it in detail. The brain is made up of glia and neurons. So neurons are not the only cells in your brain. There are glial cells, glia. And they're important for the proper functioning of neurons and for the brain's response to injury. And someone's described the glia once as a bit like the scaffolding that enables the rest of the brain to stay in place. And so glia are really important. They're a bit like the transport system. The neuron. Alright, this afternoon we're going to continue and we'll look further into the functioning of the nerve impulse. Now this will take us into some electrical and chemical information, and it will also then enable us to move into more of the geography of the brain, the gross anatomy. So we'll continue on with this microneuroanatomy till we explore the nerve impulse, and then we'll move into gross neuroanatomy. Alright, the function of the neuron is to generate and conduct electrical and chemical signals. So what it needs to do is move an impulse from one end to the other, and it does this by way of the stimulus in the nucleus and also by generating the electrical impulse through the interchange of chemicals throughout that axon that we've looked at here. So this is the nerve impulse. I'm just going to give you a moment to read that. I might just read it out aloud for you. It's a very good summary of what the nerve impulse does. The nerve impulse starts when the presence of chemical transmitters is detected from the dendrites. Now the chemicals are positively or negatively charged, so they have a little electrical impulse, a little electrical charge, I should say, associated with the chemicals. So those little chemicals are electrically charged, therefore they will help to generate the electrical impulse that moves down the axon. The chemical reaction happens that causes the inflow of sodium, which is positively charged. As sodium is positive, the inside then becomes too positive, and the layer of lipids, which is the layer on the outside of the neuron, opens up to allow more positively charged ions out. So what we've got is too much charge inside the neuron here, so it becomes too positive. Too much positive charge, and so the ions open up and allow that positive charge out, so that it equals the charge. Okay? So the goal is always to return to balance. So once that charge of too much sodium inside, because it's got too many positive charges on the little ion. Sorry?
What is it, homeostasis?
Homeostasis, yes, that's what they're trying to achieve all the time. The axon is trying to achieve homeostasis all the time. So it will open and close these little gateways to either let ions out or in. So, as sodium is positive, the inside then becomes too positive, and the layer of lipids, these little fat layers on the outside of the ion, of the axon, opens up to allow more positively charged ions out. Now, K is positively charged as well, and it is which chemical?
Potassium.
Potassium. Good. So we've got salt, or sodium, as in the Na+, potassium, K+. So K+ flows out as well as sodium. So we've got Na flowing out and K flowing out. This brings it back to equilibrium, so the process continues like a train down the length of the axon. the nerve impulse will only occur if the electrical voltage in the neuron reaches a threshold, and I'll show you that shortly. This requires a minimum amount of Na, of sodium, to flow out to begin the process. Does that make sense?
Yeah.
Okay, so let's see what this looks like. We need to, excuse me, we need to generate a nerve impulse, and that means that there needs to be enough sodium flow out, sodium and potassium to flow out to enable that impulse to reach the threshold it needs. It needs to get up to this amount. Oh, here we are, the threshold's down here. So once it reaches this, then it explodes, as it were, decreases again, and reaches the resting potential, and then comes back up to that resting potential. Now that sort of impulse moves all the way along the axon, as you can see there. Is that making sense so far? So we've got movement of potassium out, movement of sodium back in, creates the movement throughout the length of the axon. So sodium moves out, potassium moves out, sodium moves back in, etc. So it's constantly trying to achieve that equilibrium. And this is what's called the lipid bilayer. It's simply the way that if you like, the skin of the axon is made up. So it's made up of this thick layer with some cells on the outside and some on the inside. Oh, here we are, the outside is here, and the inside of the cell underneath. And these layers have constant openness, or they're closed. They're either open or closed, letting the sodium and potassium flow in or out of that layer, so they can get from the outside to the inside of the axon. These are the vital chemicals: sodium, the potassium, chloride. So you've got sodium chloride, which makes what? Salt. And we've also got calcium. And that's very positively charged. It has 2 protons. So it's got two positive charges compared to the single positive charge that sodium and potassium have. And chloride is negatively charged. So we've got different combinations of each of these will make up a balance. So the brain wants to keep an electrical balance between inside and outside of the neuron by pumping these chemicals in and out of the cell wall through these little openings constantly. So here we have a graphic of it, showing the way that the sodium and the chloride will move in and out constantly. K, the potassium moving in and out to generate that impulse as it moves down the neuron, down the axon. Okay? So, what causes, once this impulse has travelled down the length of the axon, what causes the release of this neurotransmitter? So these neurotransmitters are in what's called little vesicles or containers of the chemical, of the neurotransmitter. When the nerve impulse reaches the axon terminal, it causes an inrush of calcium. This causes the synaptic vessel to open and release the transmitter chemical. So it needs the stimulus of this electrical impulse to allow the terminal button to open up. I'll lose the words after a while. And enables then the calcium to rush in, and that releases the chemical out. So we've got a 2-step process happening Within the synaptic terminal, there are vessels or packages of chemicals ready to be released into the synaptic cleft, which is that space between the two neurons. The neurotransmitter floods into the cleft and binds with the postsynaptic receptor. That means the next neuron. This opens ion channels that permit the flow of sodium and potassium in or out of the nerve impulse, and the nerve impulse is triggered in the next neuron. So we get this constant process, constant electrical charge being used up within the neuron, within each neuron. So as you and I are functioning here, this is happening billions of times as we're sitting here all the time. There are two types of response to neurotransmitters. Some neurotransmitters will land on an opening or a receptor that it will switch off. And a neurotransmitter can also land on another type of receptor and it will switch on. So either it will be excitatory or inhibitory, depending on the type of receptor that the neurotransmitter is associated with, that it lands on. Does that make sense? So it will either close the door and decrease the chance of the next neuron firing, or it will land on the next neuron and it will stimulate that neuron to function and it will fire. So some are excitatory and some receptors are inhibitory. At an excitatory receptor, in other words, it's going to stimulate that next neuron to fire, the sodium channels are opened and the sodium ions flow in, and if enough of them flow in from enough open receptor sites and for long enough, the next neuron will fire. Inhibition occurs, as you might expect, by the ion channels that are opened to the potassium and the chloride decrease the chances of the neuron firing by creating an outflow of ions, making the inside of the neuron less positive and therefore less likely to fire or trigger a nerve impulse. So it's even, if you remember that peak, it's even further away from the threshold. It takes it down into negative territory. Do you remember that little graph that I showed you? I'll just go back a few slides. What happens in the inhibitory receptor is that it goes down here, it takes it away from the threshold level, so it becomes even less likely to fire. Here we go. I've just got one forward. Here we are. If it's excitatory, it takes it up, but if it's inhibitory, it makes it more negative and therefore less likely to fire. And this is particularly important when we think about the different types of neurotransmitters that there are. Thinking about serotonin, glutamate, melatonin, acetylcholine. So let's have a look in detail about these neurotransmitters. These are the chemicals that are released by one neuron and react with the neighbouring neuron to cause it to fire or inhibit the next neuron from firing. So if we've got too many inhibitory neurons, inhibitory receptor sites, then we're going to get too much inactivity happening. Here we have a little examination of some of the different neurons, different neurotransmitters. Serotonin, for instance, is most commonly associated with depressed mood, but it's also associated with memory, with thinking ability, with drive or motivation. And you'll notice, of course, that those are commonly associated problems with depressed mood. The depression experience is often one of not being able to think well or clearly, not being motivated to act, as well as having a low mood. So there's a collection of different symptoms that are associated with the function of serotonin. And this is why depressed mood or depression as a syndrome is not just about depressed mood, it's about all the other things that go with difficulty thinking, lack of motivation, etc. Lack of power. Speed of information processing. In other words, everything slows down. And isn't that true about depression? People with depression, they find it difficult to even speak sometimes at the normal pace. Sleep cycles get disturbed and the vegetative functions of sleep and weight and appetite become affected. And appetite depression is one of the, or reduction is one of the signal symptoms of depression. So serotonin becomes particularly important for people with depression. Dopamine. Dopamine is associated with schizophrenia. Any other connections that you can think of with dopamine?
Parkinson's, isn't it? Yeah.
Epilepsy? Okay. So, a crucial part of the experience of good mental functioning. Did anybody watch Awakening? With Robin Williams?
Yes.
Okay. The neurotransmitter there? What was the drug they were working?
Yeah, I can't recall.
Okay. Have a think about that. See if you can do an internet search to see if you can find out what was going on. Noradrenaline, associated with depression and mania, in other words, control of mood. Feelings of pleasure and pain, stress, and interestingly, learning. It has to do with motivation as well. And finally, acetylcholine, which we looked at earlier as well. The lowest levels of all transmitters in patients with Alzheimer's disease, but these new drugs are designed to inhibit the metabolism of acetylcholine. In other words, it's constant, we want to keep more of it in that synaptic cleft to enable it to be taken up by the next neuron. So it inhibits the metabolism of it, or the changing of it into something else. We've looked at that slide already. Move on. This is the process of metabolism for acetylcholine. It changes from choline and is metabolized through the acetyl coenzyme A into acetylcholine and then to acetate and choline. And that's that process that we want to inhibit so that we keep more acetylcholine available. And sometimes they're called acetylcholine esterase inhibitors. And that's what it's designed to do, is to inhibit that process that changes this into something else. These are just another graphic to illustrate the changes. We've got acetylcholine here, comes out, and we want these to be available for these receptors in this next neuron. So we want that to be all available and not turned into something else. Up here, we want it to be constantly available. And there are an enormous number of acetylcholine receptors, and what they call cholinergic pathways, in the hippocampus and the cerebral cortex. So you can see there the connections to other areas in the brain. We've got the frontal cortex, the parietal cortex, and the occipital cortex back there, and the hippocampus in here, nestled underneath in the temporal lobe. Okay, before we go into the sympathetic nervous system, any questions about microneuroanatomy? We've looked at neurons, glial cells, neurotransmitters, We've looked at the chemicals that go to make up the nerve impulse, or generate the nerve impulse. We've looked at Schwann cells. What's the name of this sheath? Myelin sheath. And can anyone explain to me how the nerve impulse progresses down the course of the axon?
Sodium and
Yeah.
potassium flows in and out from there.
Yep, in very simple terms, that's exactly what happens. It's the inflow and the outflow to maintain an equilibrium. So once it gets stimulated, you get too much sodium going in, too much gets too positive, and then it will try to re-establish equilibrium by going up. So it opens up, and it takes, potassium goes out with it, and of course then it gets too negative, so more chloride goes in, so it bring more, goes out, so more sodium and potassium go back in, so we get this constant movement in and out, and that impulse moves all the way down the axon. Okay. What's the function of the glial cells? What do the glial cells do? Transport. Good. What else do they do?
Clean up waste products.
Clean up waste products. Thank you, Johnny. They clean up the waste products. Anything else? What can you pick up there that will help us to be clear about what the glial cells do?
Okay, well I've written down that the blood-brain barrier restricts certain diseases to enter the brain,
Yes.
but will allow other chemicals to pass through to function, but also allows harmful chemicals to pass through.
Occasionally, yes.
Occasionally causing things like UTIs or symptoms of UTI.
Yeah. Good.
The filter a bit like your kidneys filter.
Yeah, it's a good way to put it. Yeah. So it allows nutrients to enter the brain, and the nutrients are in the form of sodium, potassium, chloride, calcium. So this is what the brain needs, it feeds on, and the complex mix of chemicals that help it to function. So if you think about foods that are good for brain function, bananas, we need potassium. Yeah. So, and there will be, you need a balance. So this is why good food is important for students at any time. If you think about year 12, they can't live on chips and burgers.
And the cardiac system then comes in as well, because it's really reliant on potassium as well.
Sure. Yeah, that's right, exactly. The efficient functioning of your cardiac system is going to work in close collaboration with your vascular system.
Which result into dementia.
Sure. Yeah, which comes back to your very first comment.
Yeah.
Ellie, did you want
Oh, it's a question, isn't that all related to like the cardiovascular to digestive system as well?
Sure.
So that depends what we put into ourselves?
Yep.
Yeah.
Good food equals good thinking, ability equals good living. So if we don't eat well, or if we have an impoverished diet, then it's going to show up in the way we think, in the way we solve problems, and our ability to remember, etc. Okay. So, good balanced diet. Lots of bananas.